and it will be about date vinegar, the microbiota, chemical characterization, and starter culture production. And actually, I'm going to talk about the quality and the quality also of this product. So first, vinegar is actually a worldwide product, and it is made in different countries using various cultures, and there are many varieties of it, and it depends actually in the type of the raw material that is available in that country. So the ranges of it, there are many byproducts from the agricultural low quality product that can be used to produce the high quality date vinegar at the end. The vinegar is actually have been utilized for long times, for about like 5,000 years for production of many, for a lot of usage of it. And this including using it for flavoring agent, cleaning purposes, also as antimicrobial agent and in health as well. So it is usually the date vinegar is produced from date fruit, and in Oman the date fruit, as you know, that it is very, very important crop, and if you utilize a large area that's grown in Oman. So first, the date contains lots of constituents, especially the carbohydrates, the proteins, minerals, vitamins, and antioxidants, and the fibers. For us, for the production of this product, what is important is the presence of carbohydrates. So the production of date vinegar occurs actually through two stages. The first stage, as you can see here at the bottom of this bottle, so we have here the sugar that's present in the date, and because of the presence of the yeast, it converts the sugars into ethanol. So this is the first stage of the ethanol production. And usually what I'm going to talk also about today is the spontaneous production of date vinegar. So it depends on the microflora, which is naturally present in the date itself. So first the yeast will convert the sugar into ethanol, and then the bacteria that's present there, which are basically the acetic acid bacteria, will convert the ethanol into the acetic acid into the second stage. And usually this happens through process of oxidation. Now there are many factors that affect the quality of the date vinegar. As you can see, the type and the diversity of the starter culture that is present in the date itself. So what kind of bacteria, what kind of the yeast that we have here, they will affect actually the quality of the final product. Then the quality of the raw material itself. So sometimes we have really spoiled raw material, and it's going to affect the final product. Then we have also the production method. There are many different types of or ways to produce the date vinegar. So one of them is actually the one that chooses the culture that usually that are submerged in the culture itself, and we have those that use actually the bacteria that are inoculated or they are found at the surface. So this also can change the quality of the final product. Then we have the aging, if we are going to age the product or the vinegar for longer time or not. So there are many types of vinegar. They range from red wine vinegar into we have traditional balsamic vinegar, malt vinegar, rice vinegar, and so on. And then we have also date vinegar, the one that I'm going to talk about today. So the chemical constituents of date vinegar, there are many of them. We have acetic acid, and usually according to the standard, the concentration of this acetic acid should not be less than 4%. And actually the acetic acid is the chemical that identifies the date vinegar to be as date vinegar or not. Also it gives the vinegar its tart flavor, pungent and also biting odor. What is also important in the date vinegar is the presence of ethanol, amino acids, mineral salts, polyphenic compounds, and also gluconic acid. So I got this, the gluconic acid is actually important because it also confers the vinegar greater physical stability, reduces pH, increases physical acidity. And because also in our research we found actually gluconobacter that's known to produce more of gluconic acid. Also, the gluconic acid gives the date vinegar or the vinegar itself the taste of the acidity plus also the sweetness. And this is actually preferred by consumers. So in research, we have this concern of the presence of gluconic acid or gluconobacter. Now in Oman, actually, the knowledge of the method of production of date vinegar has passed from one generation to another until we know now, especially in special festivals and such as Eid, the date vinegar is consumed, prepared at home. But this is the condition that's used for the production, and the method is actually a traditional method with there is no that much control over the production. So the quality and safety of that product is unknown. So that's actually the scientific gap that we wanted to know about it in our research. Is it actually meeting the quality and safety requirements or not? Usually the fermentation process takes about 40 to 50 days for the production of date vinegar, I mean the traditional one. And during this process, sometimes we notice that there is growth of mold and of course possibility of production of mycotoxin. The ethanol concentration might be also not meeting that standard, and the standard actually should be less than 0.5%. And then also the acetic acid concentration should be not less than 4%. So we wanted to check all of these standards. So that's why we had two projects. In the first one, we checked actually the hygienic quality of the date vinegar, and to do this, we collected some homemade samples. And then we wanted also to isolate the yeast, the acetic acid bacteria that are present in the date vinegar. Then in the second project, we wanted to formulate a starter culture to help in solving these problems. So this project is actually important because it can, at the end, provide the local industry for a scientific way how to do it and make the date vinegar in a way that makes sure that the final product is actually having high quality and safety. Also, we wanted to actually use the low quality or the less utilized dates for this production, and therefore we used the omicil variety of dates. We wanted also to develop starter culture that can actually shorten the time of fermentation. So the time of fermentation it ranges actually from 10 to 40 days. It depends on the local methods. Now the materials and methods. So we collected homemade samples, and there were 12. We were lucky actually to find something like 12 traditional date vinegar samples that were prepared at home. So we collected them, trying to target different regions. Then we prepared also laboratory-made samples exactly in the same way as the traditional method. So we tried to simulate actually the traditional method for production of date vinegar. So we produced them also in the lab, and these were the 28 samples. We did microbiological analysis for acetic acid bacteria, lactic acid bacteria, yeast, enterobacteriaceae, coliform, and E. coli to know the safety of their product. And of course, we wanted to isolate the acetic acid bacteria. And it was actually, many years until we got the best medium for the production, for the isolation of acetic acid bacteria. And in the first project, actually, we had a problem with the yeast, but then we used actually cyclohexamide to prevent the growth of yeast. And in the second project, we had another problem with lactic acid bacteria, and it was actually the time of COVID-19, and we had like 3 closes of our lab, so it was somehow difficult to do the project at that time, but we tried to manage. We couldn't get the antibiotic that kills actually the lactic acid bacteria, but eventually we found another antibiotic to kill the lactic acid bacteria and eventually to allow the growth of acetic acid bacteria. So it was actually, a long way, but at the end we had the way how to produce and isolate the acetic acid bacteria easily from the date vinegar, because the environment is really very competitive and very complex. We did identification of the bacteria and yeast using PCR, chemical analysis using to get the concentrations of acetic acid, methanol, ethanol, pH measurement, and also the concentrations of glucose and fructose. So this was in the first project, to isolate the bacteria and to know the quality of the date vinegar product. Then in the second stage or second project, we wanted to formulate a starter culture using the yeast and the bacteria that we previously isolated from date vinegar. So we use the yeast, Saccharomyces cerevisiae, in all of the starter cultures, and we use the bacteria, which are Gluconobacter and Acetobacter, for production of 3 types of starter cultures. So we have 3 types of starter cultures. We have the first one that contains both Acetobacter and Gluconobacter, and then we have one that contained only Gluconobacter, and the third one that contained only Acetobacter, of course, all of them with the yeast. Then we prepared the date broth in the first step. Here we prepared the date broth, homogenized it, and then we added the starter culture and we did the fermentation. And this time, actually, we did the fermentation only within 4 days, and not 10, 20, 30, or 40 days, to check actually the concentrations of the ethanol, acetic acid, and methanol. So these are the results from the first project. We did both chemical microbiological analysis for homemade samples. So these are the samples that we collected from local people, and these are the lab-made samples that we produced in the lab. So what we found actually that I'm going to give you only the conclusion from all of these figures. So what we have found that the local samples that were produced at home contain actually more coliforms and enterobacteriaceae, which indicate less hygienic quality than the ones that we produced in the lab. However, both of them actually, we couldn't guarantee that this process or the method of the production produces date vinegar with enough amounts of acetic acid or with a small amount of ethanol or methanol as well. So the concentration of methanol was more than the acceptable level, so it is a toxic substance. In many samples, whether the homemade or the lab-made samples, the ethanol concentration in many samples was greater than the standard level, and then the acetic acid was less than the standard level. We isolated also 5 species of Gluconobacter for the first time from date vinegar, also 6 genera of yeast. You can see here also the growth of acetic acid bacteria in this medium, that we took actually long time until we got the formulation of this type of medium, and until actually the growth of this type of bacteria on this medium becomes really very clear. So the original color of it is actually blue, and once this bacterium grow in it, it turns it to be yellow in case if it is Gluconobacter, and in case if it is Acetobacter, it will retain it back actually to the blue color after 2 to 3 days. So it is also a differential medium. So this is also one of the analyses that helped us to actually get an idea, it's what we say a component analysis, about the correlation between different factors. As you know that there are many factors that we looked for, such as the acetic acid, methanol, ethanol, yeast, acetic acid bacteria themselves, lactic acid bacteria, and so on. So somehow this helped us actually to find the correlation between different factors. For example, you can see that there is negative correlation between lactic acid bacteria and the acetic acid bacteria here. So sometimes in some samples we found that there is still concentration of acetic acid, and the concentration or the count of acetic acid bacteria was low. So because of the presence of lactic acid bacteria, actually that negatively correlated with the acetic acid bacteria, so it indicated actually their presence. So the lactic acid bacteria were present when the acetic acid bacteria were actually absent, and they may contribute to the production of the acid.
2 minutes. Dr. Zahra?
Yes. So, and this figure also shows the spontaneous production of date vinegar from two samples, sample 1 and sample 2. And actually what I want to show you here, the production of high concentration of acetic acid by the spontaneous fermentation. So this is actually the greatest amount that I could find actually in the literature. I couldn't find any production of lactic acid, of the acetic acid that reached to 10% like this one. So this was from our second project. And here you can see also the presence of the acetic acid bacteria, the top layer here, and also in the transplant. And these are actually the bacteria, the acetic acid bacteria. These are the types that we isolated from different types of bacteria. It wasn't easy actually to get them, but at the end actually we got 3 different species also for the first time from date vinegar. They were Acetobacter malodorans, Acetobacter tropicalis, and Acetobacter persistens. We used these to formulate and produce this vinegar within 4 days. So we were able actually to reduce the time of fermentation from let's say 10 to 40 days to 4 days. And the concentration of the acetic acid, as you can see here in the samples that contain especially the Acetobacter, there were enough, they were acceptable, more than 4%. So the recommendations and future research needs for this project, so the way that the date vinegar is produced cannot actually guarantee the production of enough amount of acetic acid. The ethanol concentration is greater than what's the recommended level. The methanol, which is toxic, is also present there, and it is more than the acceptable level. Also, it may contain some bacteria that are pathogenic, or coliform, they were found as well. So that's why it is really important to control the production of date vinegar. I would like to thank SPU for providing funds for this project, and also to thank the research team, Prof. Najib, Dr. Ismail, Prof. Abdullah, Dr. Nasser, Ms. Fatma, and Zainab as the research assistant. So far we had one published paper and another one under review, and you are welcome to ask questions.